We are live. I'm going to share my screen.
Okay. Hi,
Okay.
everybody. Marine, can you just wait a bit?
Oh, yes.
I'm gonna first leave the ground to and the floor to Andrea, who will be the chair. So first of all, hi everybody. We're back for this nice session of coded tools, and I leave the speaker and the microphone to Andrea Brovelli. Please enjoy.
Okay. Hi, thank you, Clément, and congratulations to the organizers because you've been doing a great job in organizing this nice workshop online, which is very, very challenging. So the session is a coded tools. The objective was to present new softwares for to improve the research practice in EEG and MEG analysis. And so we will have three speakers, and the first one is Marijn van Wit, who's a postdoc at Aalto University in Finland, and he studies language processing in the brain. And in addition, has been publishing several articles about reproducible science and reproducible analysis pipelines, and he's also one of the developers of MNE Python. And today he will present us a seminar on how to design analysis code that scales in space, time, and I leave you the word.
Okay, thank you very much. Yeah, let's dive right in. So welcome everybody to designing analysis code that scales. And I'm going to do something very challenging. I'm going to analyze some EEG data, and I'm going to do this live because I want to show you my workflow, how I process these data analysis pipelines. And this is a workflow that's been working very well for me. So hopefully you can pick up some useful tips. I've prepared some EEG data for us to analyze. This is the brain invaders data set. It's a P300 data set where subjects were looking at a screen like this, and there's these space invaders, and you would look at one of these space invaders. And whenever your space invader would light up, you can see in your EEG signal, you see this P300 potential. And I'm hoping that many of you will know about this already. So let's see if we can spot this in the EEG data. Now, I don't have enough time to really do this all the way from scratch. So I've taken the liberty of already populating my folder with the study template that you can download from our GitHub session. And I already wrote two analysis steps. So I have one analysis step that will download the data and one that will pre-process the data. And you see in my workflow, every analysis step gets its own Python script, and they are numbered in the order in which they need to be run. So the data is now ready for us to start cutting epochs. But before we can write the third analysis script that does that, I want to show you the config file. So the config file really is the heart of my data analysis pipeline. This is where all the important parameters get defined. And this config file starts by checking on which system the analysis is running. So if it's running on my laptop, this is where the data is going to be. If it's running on my workstation, this is where the data is going to be. If you are a collaborator of mine, you can add your own system to this list, and the same code will run on my machine, on your machine, everywhere. That's very useful to have a construct like that. Then, of course, come the all-important parameters. Now, for a parameter to end up in the central configuration file, as opposed to in one of the data analysis scripts, there can be one or two scenarios. So either the parameter is used in more than one script, or this is a parameter that's important enough that you want to report it in the method section of the paper. We will see how that works in a moment. So right now we only have the parameters here for the pre-processing step. The filter cutoffs are there. And finally, we have the names of all the files that are relevant for our analysis. I collect all my file names in a central location, which is inside the config file. And I use this neat utility class, this file name utility class. So here I have a Python open, and let's see how that works. So for example, if I want the URL where the data can be found, this is defined here, and this has this little subject placeholder. Can you see that? So when I want the URL, I can go fname.url, and I can say, well, I want the URL for subject 1, and there we have it. Or I want it for subject 15, there it is. This is super useful. You will see how useful this is. Please do this. Right, let's dive right in. Let's go. Let's create our third analysis step, cutting epochs. When you start a new analysis script, always first write a comment. What is this script going to do? It's going to cut epochs, and it's going to compute evoked potentials. Always document your scripts a little bit like this, like what is this script doing? We're going to use ME-Python. Well, I'm one of the developers. Of course, we're going to use ME-Python. And from our central config file, I'm going to need this fname utility class. So let's get started. First, we load the preprocessed data. So we can just load it like this. So now, file name object, give me the file name where the filtered data lies for subject 1, please. Yeah, go ahead and preload that data as well. Okay, let's see, does this run? Yeah, it runs. Okay, we're up to a good start. Next, we find events. Right, so we start writing the script. We're going to find the events on the filtered data, and our stim channel, the name of the stim channel is target. Right, is that running? That's running. Okay, now we have events, and we have 2 events. We have an event that's generated whenever a space invader is flashing that we were not paying attention to. That's event code 1. And whenever a space invader was flashing that we are attending to, that is event code 2. Let's see. Well, let's cut epochs from a little bit before the onset of the flash until like 1 second after. That should be nice. And we're going to use this pre-onset time as our baseline. So now we can define our epochs. So I'm sort of assuming that you have done this kind of evoked analysis before. Otherwise, I'm afraid you're going to be a bit lost. But just bear with me. Okay, so here we have our epochs. Let's run this. Yes, we have them. We have almost 1,200 lovely epochs being cut. Lots of non-targets, some targets. Great. Hey, but these 3 parameters I now define, these are the kinds of things that you would report in a paper, isn't it? These parameters need to go into my method section of my paper. So I'm going to take them out from here, and I'm just going to put them in my central configuration file. So all the parameters that need to go in my method section, I'm going to collect them and here in a centralized location. So I have all of them, all of the stuff I need to report, I have them in a single location. Okay, now of course Python's going to complain that it cannot find them anymore, so we'll import them right back into this script. Check, is it still running? Yes. Great. What's next? Evoke potentials. Okay, let's go. Evoke potentials for the targets. So we take all the epochs for the target event and we average them. Average. And we do the same for the non-targets. Right? Okay, is that running? That's running. Great. Final step, save everything. Let's save our epochs. And file name object, give us a suitable name for the epochs for subject 1, please. And go ahead and overwrite them. And we're going to do the same for our evoked, obviously. So fname object, give us a file name for the evoked for subject 1. And there we're going to target and our non-target evoked. We're going to chuck them in there. There we go. Is that running? No, it's not running because we actually need to define these file names. So here, our giant list of file names. We have 2 more for you. We're going to have the epochs file name and the evoked file name. And I'm going to name the epochs the epoch and Ava. So these are sort of the template file names that ME-Python uses. Does it run now? Yes, now it runs. No, it doesn't write evoked. No, it writes evoked plural. Wonderful. Okay, now I have them. Let's see if we can visualize them. So our target one, let's see, did we get anything? Yes, we do. So these are our evoked potentials for a single subject. And you can clearly see this P100 effect. Lovely stuff. Okay, that's our evoked analysis for a single subject. But we have like 60 subjects. Am I going to write a for loop now for subject in subjects? This, and just do this for all the subjects. This would be a horrible idea. Because now, as soon as I write this, I lose the ability to run this script for a single subject. I've been doing that all the time when I was writing this. It was super useful. As soon as I write this for loop, I cannot do that anymore. So I'm not going to do that. Well, I need that line. Let's go back. Instead, we're going to bring in the Python's argument parser. Argument parser, come here, you. And I'm going to define a command line parameter. So this script is going to get a command line parameter, and it's going to be called subject, and the type is going to be an integer, and this is going to be the subject to the process. It's going to be one of like 71 subjects that are defined here. So now go ahead and actually parse my command line arguments into this args structure. So now instead of subject one, I can just feed in args.subject. So now it will analyze the whatever subject we specify. So if I now run this on the command line, it's going to complain at me saying, no, I need a command line parameter. I need the subject. So let's analyze subject number 2. There it is, or 15. Yeah, so we can now analyze any subject that we want. That's amazing. Okay. Now, we need to run this for every single subject. I'm not going to do that by hand. I'm going to bring in a build system. This is the PyDoIt build system. I love this little build system. And what it allows us to do is to define tasks. And you do that by writing Python functions. So let me define a new task here, the epochs cutting task. Give it a short description. So this cuts epochs and computes evokes. And actually, I'm going to define a subtask. So this big task will have one subtask for each subject. And I do that by yielding dictionaries. And the subtask would need a name. And so I just give it sub and then the subject number nicely formatted. And now I'm going to tell PyDoIt for this task what files are required to execute this task. Well, I need to filter data for the subject we're currently doing. And I need that epoch script that we just written, right? Those are the files that we need. And now I'm going to tell it which files are going to be produced by the task. Well, we're producing epochs. And we're producing evokes. See how useful this fname like little object is. I don't have to think about file names and I just write them as I go. Super fast and easy. Okay, and now what do you actually need to do in order to do the task? Well, you need to run Python on this lovely script, if you like it, and give it the subject as a command line parameter, right? Okay, so now I can go to my DoIt tool. So if I list the task, it will show me I have 3 tasks defined now. And I can run a specific task, but I can also just type DoIt and it will run everything. And it's going to be clever about it. Look, it already knows, it already detected that we have downloaded all the data already. We already pre-processed the data already. So all these steps can already be just skipped. And we can jump right into the computation of the epochs. It's now computing epochs for all my subjects. But it's very slow. So I'm going to cancel this. I have 6 cores in this laptop. So let's process 6 subjects at the same time.
You have a couple of minutes.
Okay, yes, a couple of minutes left. Excellent. So now it's running very quickly. And we already skipped the few epochs that we already computed. Okay, and while this is running, let me finish up quickly by writing final script that computes the grand average. And for that, I need the file name object, and I'm going to need my subjects because in this script I am going to loop over my subjects for subject in subjects. Okay, so you're going to read my evokes, evoke file name, and the condition is target. And we're going to read the non-target. And we're going to add these to the lists. Oh, we need Python. We need Python as well, right? Always have a linter running along when you're typing, right? Excellent. And when we have those, we can compute the grand average. So that's the grand average for the targets, and that's the grand average for the non-targets. Lovely. On the left, I can see my data analysis is done for the epoch. So let's see if we can compute grand averages. And we can, ladies and gentlemen. And now we can visualize them. So give me the grand average for everything. And let's see. We have a wonderful P200 potential there. Right, so that is my general workflow, right? See how fast this is and systematic this is. So here are our 4 analysis steps, and they're just clear here. We have a build system that will tie everything together. It's a very systematic way of working. So I hope this was useful to you guys. And you picked up a few nice tips that you can employ in your own data analysis pipelines.
Thank you very much, Marijn, for this amazing performance. I think if you had another 10 minutes, you could have written the paper. Very nice.
I think so.
And,
I want to do this, but yeah.
I don't see any questions from the audience. Maybe I can ask a couple waiting for some questions from the audience. So I've seen that you have a paper that came out recently in March, which is entitled 7 Quick Tips in the analysis of scripts in neuroimaging. How that paper, and so I've seen that you have implementing different pipelines for the analysis. Can you give in 1 minute like a hint of what are the types of analysis that you can do? And how people can actually share, get to know, use this code?
Yeah. So the paper we're talking about here now is it defines 7 guidelines to how to structure your data analysis scripts. And if in this little demo I've shown almost all of them. They're already incorporated here, so you can read them there. So if you want to get started with that pipeline, so you can already go to my GitHub page, and you can download the study template, which will like set up this whole framework that I have been using right now. And of course, you can do that and read through it like in your own time carefully.
Excellent.
You can start using this.
Okay. Yeah. Can you then post your GitHub page? I just put the paper on the chat.
Yeah, I will put it in the chat. Okay.
Yes. And last questions. Then we pass the word to Lawrence. What would you be able to then merge different softwares using your approach? With that generalized, yeah.
Not easily. I mean, you can merge them with PyDoit, but my own approach, so the way I've now fine-tuned it is really like just staying in Python. If you're going to merge different tools, I would like to highlight this NiPipe project. So that's the domain of NiPipe.
This is what I had in mind. Yeah.
Yeah.
Okay.
That's great. Yeah.
Thank you very much, and thanks again for the presentation. Yeah. And now we go to Lawrence Kroll, who is presenting next, and he will be on live soon. So I can start presenting him. So Lawrence is actually finishing his PhD apparently this week. So good luck to him. And his PhD project is on EEG-based brain-computer interfaces, and he's working at the Berlin University in Germany. Here he is. So Lawrence, he will present a recent toolbox for the simulation of EEG event-related potentials. And please go on.
Thank you very much. Let me see to share the right application. Okay, I think this should be the one. Thank you, Andrea, for the introduction. I would like to correct that this is not just about event-related potentials, but any kind of event-related activity. So for next 15 minutes, I will be talking about how you can simulate event-related activity using, in this case, a toolbox called Siriga. Of course, the first question you may want to ask is, why do we even want to simulate data? And I can think of two possible reasons to give you for that. The first is to test and evaluate EEG processing methods and analysis pipelines using a known ground truth. Because with simulated data, you know exactly what is in your data. You have complete control over this data. So you know exactly what the results of your analysis should be, or the result of your analysis pipeline. So with simulated data, you already know the result of the analysis before you run the analysis. And so you can really validate and evaluate your methods and pipelines. So when we're talking about good scientific practice on this conference, it is good scientific practice to validate the methods before you publish or use them. Another reason is very simple. Simulating data is cheap. You don't need to invite 20 participants to your lab, put the EEG cap on, and sit 3 hours on a chair while they do the experiment. Guess we all know how it is. If you just need some data, and this happens, it's much easier to just write a few lines of code, and there you have it. So this is something that we can all use every once in a while, just some data. And simulating the data is easier, especially when you have a toolbox like Siriga. But how would you simulate? Of course, there are different ways to simulate data, but there is one that is particularly prominent these days, that's been used in the past, and this is also the same one that Siriga uses at this moment. It relies on an assumption that you may recognize from blind source separation, for example, where scalp EEG is assumed to consist of the summed activations of localized sources projected onto the scalp. You may recognize this formula here. So we assume that the brain basically consists of these localized sources that are all doing their own thing, their own neuroelectric thing, creating activations. And then these are all projected onto the scalp, and at the scalp they are summed together and recorded by the EEG electrodes that we have. So how do you simulate data? You define these localized activation patterns, you project them, sum them together. That's it. So if you want to have X, which is the EEG data, you need A and S and maybe the epsilon in this formula. Of course, there's a bit more to it than that, but this is the gist. And like I said, this is how also Siriga does it basically. So let's talk about Siriga. Siriga, it stands for Simulating Event-Related EEG Activity. It is a free and open source MATLAB-based toolbox to generate simulated data mimicking event-related EEG activity. And here's the Journal of Neuroscience publication that goes into more detail. I have put here the GitHub link also. And please do not hesitate to open any issues on GitHub if you have questions or improvement requests. And finally, you can also download it using the EEG Lab plugin manager. The general idea of using Siriga, if you want to simulate some data, there are 4 steps basically. You start with a head model or a lead field. Then from within this head model, you select your individual sources, so these localized brain sources that you want to have in your separation. Then you define the activity of these sources that you selected. And then finally, you simulate the activity for the scalp EEG. So let's quickly go through these 4 steps. As I said, the first step is to obtain a lead field or head model. This is basically the virtual setup for your simulation. So it contains the virtual brain that you're going to use, but also the electrodes that you're going to use. Siriga at the moment supports about 5 head models, but this can be extended quite easily. And you can have different reasons to go for one head model or the other. If you don't have any specific reasons, I would recommend to start with the New York head. That's the one on the left here. So let's look at some code. I mean, that's the session we're in, right? Obtaining a lead field in Siriga is actually very easy. It's just a single line of code. So LF generate from NY head would give you the lead field from the New York head. LF stands for lead field. At this point in the same line, you can also indicate then which channels you actually want to have, or you can also use a pre-specified montage of channels. And of course, there's some plotting functions. So the previous slide, those pictures were also plotted using Siriga. So we have our virtual brain, basically. We have our lead fields. But what is this thing really? One way to look at it is that it is basically a selection of all the virtual sources that are present in this virtual brain. So what you can do now is just select one of all these sources, multiple, and just assign some activity to them. So you can put an event-related potential in one source, you can put some noise in another source, you can put some alpha band activity in another source, and so on. So the next step is to actually select these sources that we want to have. There's two parameters here that are potentially relevant. First of all is the location of the source, so just the XYZ coordinates here on the left, and also the projection of this source onto the scalp. So these sources are simulated using a dipole, and this dipole can be oriented differently in space. So that is illustrated here on the right. And this is one of the nice things of using the New York head instead of the other currently available head models. The New York head comes with a default projection, default orientation. So actually you can ignore that when you use the New York head. Again, some code. This can again be one line of code to get the sources you want. There are different options to do it. You can get a random source. You can get a source at a specific coordinate or nearest to a specific coordinate, depending on what's available in that model. You can get all the sources from a specific point or coordinate in the head, or you can constrain specific sources to be a certain distance away from other sources that you have selected. And of course, there's some plotting functions as well. So at this point, we have our virtual brain, we have selected some sources, and like I said, the next step is to assign some activity to these sources. Out of the box at this point, Siriga has the following activations to choose from. There's the ERPs, the event-related potential, so just the systematic deflections in the time domain. There are the ERSPs. This is basically any kind of oscillatory activity, either constant, single frequency, multi-band, and modulated or not. So synchronization or desynchronization, that's all in this ERSP class. There's noise, so this is just like the 1/f noise, like brown noise, pink noise, and so on. There's also a class to use autoregressive models to generate some activities. This can be used to simulate functional connectivity between different sources. And again, so Siriga is modular. So if there's something else, you can just write one or two scripts and you can add it to this list. Alternatively, there's also a way to just include external data in the simulation. So if you just have some time series yourself, either something you simulated or actual data that you recorded, you can include that as well. Let's use the ERP as an example. We're going to simulate data with an ERP. In Siriga, the ERP is just simulated as a deflection in the time domain, like a single peak, as I said, which has a latency, an amplitude, and a width. So in MATLAB, you would define a struct that has this information. So here we have one ERP that has two peaks, one at 450, one at 550 milliseconds, a width of 200 and 200, and a peak amplitude that are inverted for the two peaks. Now, Siriga simulates event-related activity, as I said. So you simulate multiple epochs. If we were to now run this simulation with 100 epochs, you would have 100 epochs with exactly the same ERP. This is, of course, not very realistic, so Siriga includes a number of additional parameters that allow you to add variability to the other parameters. So either trial-to-trial variability or variability over time. But I'm not going to get into that right now. But it's important to realize that, of course, to get realistic data, it needs to be, it should not be static. But this is how you simulate or define activity in Siriga. You use a struct and you put all the parameters in there. So you say this is an ERP and this ERP has this a peak at this latency and this width and so on. And it works the same for noise, these ERSPs, and any of the other classes that I just mentioned. So at this point, we have our virtual brain, we have our electrodes, we have the selected sources that we want to use, and we have our ERP. What we haven't done then is put them together now. We have a separate list of sources and a separate list of like variables with our activations. So we want to combine them. In Siriga, this is called a component. where you put these two together. So here we create a component using the source SRC and the ERP we just defined from the lead field. And you end up with a component that then has like a location in the virtual brain and a time series activation. And this does not have to be in one-on-one mapping. So you can have one component with multiple signals. So here we have the same source using or adding the ERP plus some noise. In this case, brown noise. So this looks a little bit more realistic. That's clean. So that's basically it. That's all we need. Now we just need some final parameters, which is again using a struct. So let's say we want to have 100 epochs simulated. Each epoch should have a length of 1000 milliseconds, and it should be 1000 hertz. Then we just ask Sriga to generate the scalp data, and that's it. So just to summarize, we have here in, 1, 2, 3, 4, 5, 6, 7 lines of code. We have basically simulated an entire data set. We start with some simulation parameters, the epochs. We get our lead field from the New York head in this case. We get a source from a particular location. We define an ERP and some brown noise. We add the two together. We simulate the data. And this is what that would look like. We're already done. Of course, what I just did is really just a single component, a single source with a single ERP and some noise, which is not very realistic. You know, EEG has many, many sources. But this was just to illustrate how easy it is to get going. With just a little bit more effort, so in the paper that I mentioned before, I only have 5 ERPs. So it's basically a little bit more effort than what I said before, but not much. Just 5 more lines of code. And with that, you can actually achieve quite realistic results, in this case, simulating visually evoked potentials and P300.
Lawrence, I think you should try to wrap up, please.
Yes, I'm almost done. I just want to briefly show this impression. It is not just coding. There is also a GUI, a graphical user interface, that is available from within EEG Lab. It makes some things a little bit easier, but of course you have the most flexibility when you use the scripting. So to wrap up, data simulation can help you evaluate to validate your EEG processing methods and pipelines. Siriga is one way to do that. At the very end, I would like to mention that data simulation in general, and Siriga in particular, is of course just a tool, just like any other tool, and it has broad capabilities and certain assumptions. So it is up to you to make sure that the data that you simulate is actually appropriate for the purposes that you're simulating it for. Thank you very much.
Thank you very much. So we have time for 2-3 questions. So there's been some activity now in the panel of questions and answers. 7 votes for the question to Lawrence. How do you compute lead field? Which method?
So the head models that I mentioned, those are pre-computed lead fields. Well, not all of them, but the New York head is a pre-computed lead field. So this uses fMRI data, and it has just computed a neurophysiologically realistic lead field. So this makes it easy. We just use something that somebody else computed for us, and we use it in Siriga.
And can you import? Yeah, very good. And can you import also head models, I guess?
Yes. So first of all, to finish the last question, you can also compute one using FieldTrip. So FieldTrip is supported in Siriga. That allows you to calculate your own lead fields. But yes, it is quite easy to add another head model or lead field to Siriga.
Okay, very nice. Another question is, what would be your recommendation and advices to simulate intracranial signals recording using ECOG or SEG?
I guess so the lead fields that are being used here, they are really for EEG. So they have electrodes on the scalp, and then they simulate that. I think the first step, I'm not an ECOG expert, I have to say, but the first step would be to actually have either then a lead field that is specifically calculated with electrodes at the brain, the cortical surface. Yeah, the rest, I'm sorry, I do not know enough about ECOG. If perhaps other processes would have to be finished, have to be changed. But this is at the moment aimed at EEG.
Okay, thank you. Maybe a last question, a very general question, but then we will have a discussion roundtable later so that we can probably discuss further questions that were presented. What about integration then with MATLAB-based software such as Fieldtrip, EG and Brainstorm? How far have you gone?
So as I mentioned, this is actually an EEG plugin, or at least available as an EEG plugin, and it does also use some EEG functions. So the integration with EEG is there. Fieldtrip is used by Siriga to, if you want, calculate a specific lead field. So it allows you some more flexibility to get your own head model in there, your own electrode positions, and so on. Brainstorm, I am not familiar with, so I cannot answer to that.
Okay, thank you very much. And for those in the audience, please keep your questions for later, that we'll have some time to discuss. Now we have 5 minutes break and we'll be back at 5:15. See you later. So we are nearly back. So if you can put Etienne Combrisson on stage. I can start introducing him. So Etienne Combrisson, is a postdoc. Here he comes. Here he is.
Hello.
At the Institute of Neuroscience of Latimon. We are working together in the same group. And he's working on the neural basis of human learning. And recently he's been developing a toolbox to combine information theory and group level single subject statistical inference. And so please welcome Etienne. Now you have 10 minutes, and then please leave a couple of minutes for questions.
Yeah, okay. So thank you, Andrea, for the introduction. Is it full screen? It's okay.
Yeah.
Oh, okay. So my name is Etienne Combrisson. So as Andrea said, I'm working in the BrainNets team here in the ENT, and I've been working on a software that is called FRIT, for Framework for Information Theoretical Analysis of Electrophysiological Data and Statistics. So it's a pure Python software that we started last year in 2019. And so here is the link if you want to download it. And there is other software that are developed in the BrainNets team. And it also comes with an online documentation and examples that are built with the Sphinx and the Sphinx Gallery. So if I had to summarize FRIT into 3 key points, I would say that first, it's really made for electrophysiological data, so EEG, MEG, and also intracranial data, which is really important for me since I'm working with intracranial data. The second key point will be to be able to characterize and to extract cognitive networks that are task-related and using the information theoretical framework. And the third key point would be to be able to do inferences both at the single subject and at the group level. So just a quick example of what I mean by task-related network and the different level of analysis. Imagine you have a 3-node problems with 3 brain areas like the ACC, the insula, and the VMPFC. And in your task design, you have 2 conditions, a condition A and a condition B. The first question you can ask is, can I find some brain areas for which the neural activity is modulated by the task? So in this example, you can see that X and Y, both nodes, have an activity that is different according to condition A and condition B. The next level of analysis is a little bit more ambitious. In this case, you compute the undirected connectivity between all of the nodes, and you try to infer if this connectivity is different between your 2 conditions. And the ultimate goal that we try to achieve with FRIT is if we can be able to extract information flow from one brain area to another and see if this information flow is also modulated by the task and is different between the 2 conditions. So that are the 3 levels of analysis that we try to achieve with FRIT. So as I said, we are using the information theoretical framework, and in particular, the mutual information, which is such a versatile tool. Basically, it measures how much 2 variables are statistically dependent. So it's a measure of effect size, which means that the more dependent your 2 variables are, the larger the mutual information is going to be. And if you want to read a bit more about information theory, I strongly recommend the paper of Robin Ince. It's a really nice paper, and he also provided the code, Python code and MATLAB code of a special type of mutual information that I think it's originated from the economy. It's called Gaussian copula mutual information. It's a binning-free method. It's really nice. So here in this presentation, I'm only going to scratch the surface of what you can do with MI. So the first case, first type of analysis that you can do is what we call a discrete case. It means trying to contrast the brain activity that comes from two or multiple conditions. So for example, you compute the mutual information between your brain activity that are coming from two conditions, like condition A and condition B. So in a way, this is really similar to what you can do with machine learning, for example. It's really the same syntax. And a continuous case, where this time you try to find which brain region correlate with a continuous variable. So this continuous variable could be, for example, the surprise or a prediction error, a learning curve, something that emerged from a behavioral model, for example. Then in FRIT, we also try to be able to draw statistical inferences at different levels. So the first level is at the single subject level. So in that case, the mutual information is computed for each subject, and we detect in one single brain area and during a particular cluster of time points how many subjects does have the effect. For group level analysis, the first one is a fixed effect, where this time the data of the several subjects are concatenated. So this is really like considering that the data are coming from one single subject, which means that you make the inference, you make the hypothesis that your effect should be quite consistent across your subject. But it's a really sensible method. But however, you can't generalize to new subjects. So that's why we also have a random effect analysis, where this time the mutual information is computed for each subject, and then we make the hypothesis that the mutual information across subject is normally distributed. What is nice with that is that, in fact, it's like considering that your small population of subjects has been randomly picked from broader population of subjects. And if you compute the MI on this broader population, it should fall inside your model. So that's why you can say that you can generalize to new subjects. However, one drawback is that to build this model, it usually requires a higher number of subjects. For the p-values, we used a non-parametric statistical test using permutation, where the relation between the brain data and the continuous or discrete variable is broken. So it's basically a swapping across trial. And for correcting for multiple comparison, we use the cluster-based statistic, but we also have maximum statistic and FDR. So a small, really minimal example of how it works inside FRIT. So let's say you have the data from 3 subjects, and you put all of your data inside a list, and we are supporting NumPy array, MNE types like MNE epochs, for example, and also data array from Xarray. If you don't know, this is, to me, it's really a fantastic library to work with, especially with intracranial data. Then you also concatenate your vector of condition or your behavioral vector for each subject, and the anatomical information, because FRIT also uses the anatomical information to be run. So then you use this dataset FE, which is a container for multiple subjects, and you import this class, which is called WFMI, for workflow of mutual information. And if I had to say, I would say that this is really the core of FRIT. This is really the function that we use to infer task-related changes. So you can specify if you want to perform a fixed effect analysis, random effect analysis, what kind of mutual information you want to perform. If it's between continuous brain data with a discrete vector of condition, or if it's CC for a continuous variable. You can also do CCD, where you compute the mutual information between your brain data, a continuous regressor, and you remove the influence of a discrete variable. And then you run the workflow, which can be run in parallel. The permutation can be run with parallel computing with JobLib, and you get an estimation of the mutual information for each brain region and at each time point. and the p-values that are associated corrected for multiple comparison with the exact same shape as the MI. So now we are really in the stage of trying to bulletproof it and see if it works and if we are able to reproduce some results. Here is some data I used during my PhD. So it's a center out motor task with intracranial data where the subject is moving the hand, the mouse into 4 possible directions: up, right, down, and left. And during my PhD, I used machine learning to see if I can find the significant differences in the high gamma activity of this premotor site, and I was able to replicate it with the mutual information where you see a significant cluster of MI that is occurring when the subject is moving. The second task I'm going to present right now is a visual motor task. It's MEEG data. So in that task, the subject is learning to associate finger movements with color that appeared on the screen. So there is a first, in the early trials, the subject doesn't know which finger movement correspond to which color. So he really has to explore what is the good combination to do. And once he learned it in the late trials, he's really exploiting the finger association to move to the next trial. So the question you can ask is whether there is a difference between the exploration and exploitation phases. And we found that we replicated results where we found strong differences between exploration and exploitation in occipital regions. So now with FRIT, we are progressively moving into the direction of connectivity, since all the slides before was really performed at the node level. And we started to include several connectivity measures and compare them. So one I'm going to present right now, it's called dynamic functional connectivity. Basically, it's computing the mutual information between the brain activity of one region with the brain activity of another region inside a small temporal window. And then you can use a sliding window to have some kind of temporal resolution. So it's undirected measures. So that's why the matrix is symmetrical and it's pairwise connectivity. And we also started to include a measure of connectivity that Andrea have been working on. It's called the covariance-based Granger causality, where this time there is this notion of information flow and of directionality from one source in the past, and that causes an influence on a target in the present. And you know that by conditioning by the past of the target to be sure that this is not the past of the target that is modulating the present. So that's why you have a directed connectivity, non-symmetric connectivity array. So what is nice with both measures is that they are performed at the single trial level. So which means that you have one connectivity array per trial, and you can compute it for all of your subjects. And if you have, for example, your discrete vector of conditions, where you have condition A and condition B, you can try to compute then the mutual information between your undirected connectivity array with your vector of condition, and see if there is...
To wrap up, please.
Yes, and see if there is task-related differences in connectivity between the two conditions. One further step would be, instead of using directed connectivity, using directed connectivity, and see if you have differences in directed connectivity between your conditions. The ultimate things would be to find directed connectivity that correlates with a brain model, for example. And with still this possibility to perform inferences both at the subject and at the group level. Okay, so this is my last slide, just to give you again the link to the software if you want to try, and just a word to say thank you to the people I've been working with. So of course, Andrea, Robin Ince, which helped me really a lot, so thank you, Robin. Bruno Giordano, Michele Allegra, and Ruggero Bassanici from the INT. So thank you for your attention.
Okay, thank you very much, Etienne, for the presentation. I think we have time just for one quick question, then we go to the round table. There's one question that has been voted 3 times for you. Now I get it back. So, does the Gaussian copula mutual information method make Gaussian assumptions about the data? And do we need to do any transformations to achieve these assumptions? And can we use source localized EEG data?
So the answer is no. It doesn't make assumption of Gaussian variables. However, you make the assumptions that the relation between your 2 variables is monotonic. So, or no, not monotonic, but roughly monotonic. It's not really strict, I guess. Yeah, that's why it's called Gaussian copula.
And so do you need to do any transformations to achieve this?
Yes.
And can you apply it to source level EEG data?
So what do you mean by transforming? Making it Gaussian, like Z-score? No, you don't really need to make your variable Gaussian. I'm working with SEEG data without any normalization, and it works quite well.
There is the COP norm normalization.
Yep. But it's really for extracting the copula. It's not really, it's inherent to the Gaussian copula method. It's not really transforming your data.
Okay, thank you very much, Etienne. So now we have the last 15 minutes to have a round table. I think maybe if you can put the screens of all of us, so the 3 of us, or the 3 speakers. Yes. So Lawrence is coming. So we don't have. Okay, so there was, maybe we can ask some questions that have been voted a lot in the chat that were not addressed during the question time, and then maybe we keep like 5 to 10 minutes to make a round table. So there was a question to Marijn, which was about, do you have any recommendations to automatize or optimize the pre-processing with ICA?
Yeah, so all my ICA analysis are, of course, automized and pre-processed, and it just follows the same pattern as I've been showing. So you will create a new script, a new step of your analysis. You call it ICA, and there, I mean, MiniPython implements all the ICA things that you would need to do this. So you just write a separate ICA script to do them in, and then, I mean, a complete tutorial on how to do ICA in a complete reproducible way is a bit out of scope, I guess, for a response for a question like this. But the general answer would be that yes, MiniPython can do it. I think I already responded to the question with a link to a paper, a tutorial paper that explains that goes over exactly how to do it. So that should be a good start.
Okay, thank you for this. So I think we have to go pretty quick. Maybe we can spend the last 12 minutes trying to really do a round table. And I actually selected a couple of topics that I would like to have your opinion and on which maybe you could try to converge. The first one is a problematics which is related to how to test methods for, say, functional connectivity analysis or source level connectivity analysis, and the use of simulations or scripts for simulating network level data. So in Fieldtrip there are some functions to do this, then we've seen from Lawrence that you can use this and you can integrate it into softwares to project the data at source level, maybe then to reconstruct the source level. But what are, do you think, from your point of view, the state of the art? Do we need these tools? Do you recommend these tools, and what are the best software to do this? So maybe Lawrence, can you start?
Sure, I can mention a little bit. So the way which functional connectivity is implemented in Siriga at the moment goes back to a implementation that was first done by Stefan Haufe. This is now 4 years ago. They published a simulation framework for connectivity measures. So they have simulated in a similar way as now Siriga would do a benchmarking data set, basically to compare different functional connectivity measurements. So that would be one way to go about it, to have like an objective data set where you know exactly what the functional connectivity is, and then you can apply different connectivity measures to see to what extent they can recover the ground truth connectivity.
Okay. And about the type of models for each node, what would you suggest? Regressive models approach, for example?
That is the one I have at the moment, yeah. So this is, to be honest, this is something that I copied from the state of the art at that time. I have not updated my own knowledge with respect to functional connectivity, so I'm not sure if there are better methods at this moment.
Okay, thanks. Marijn, what's your view on this? And do you have also ideas about how you can create automatized pipelines for this?
Yeah, well, I'm very happy to hear from Laurens that there is simulation code available to simulate this connectivity things, because I've worked on the ComPy package, which is a package for doing functional connectivity, like coherence-based functional connectivity on MEG data. And a big part of that, developing that, was indeed trying to get a simulation up and running in order to test whether the methods actually worked and if everything was implemented correctly. And this was super important, and this was not easy to do at all. So I'm very happy to hear that there is like software to generate these things. As far as I was aware, so at least Python doesn't supply this. When I tried to do this, it was still a pain. So if there is now software, great.
Nice. Etienne, what is your view about this? How do you test your methods?
Well, I usually generate some really dumb simulated data at the beginning, and then I progressively move to more sophisticated data. I think there is a really nice paper of Joseph Lisier about the ID TXL package, where they simulated 100 nodes and then use its ground truth connectivity array, and then they used statistical measures like sensitivity, specificity, recall, and measure in terms of statistics how much which method is performing. That say it's still on simulated data, so I don't know how much it transferred then to real data context. I think it's really
Okay, can you then post the paper so that people, Joseph is here, so that we can have a look?
Okay.
And do you have any experiences, or have any of you trying to use more biological realistic models, such as, for example, the one I have in mind is the virtual brain project that tries to combine not only functional connectivity or connectivity-based measures and models to project and to create data? Are you aware of these, and do you think it's a way where we should go or not for in practice to analyze cognitive-based EG and MEG data?
So I will take that question.
Yeah, good.
So the way I see it, or the way I was treated it, is that the simulation is just a verification of your method, because you control the simulation always, and you always simulate the data in such a way that the assumptions of your analysis whole. So if your method has a linear assumption, like we saw before, that okay, we take those linear summation of some dipole sources, then in my opinion, this is, if you want to test that method, this is how you should simulate it, by placing the dipoles and making a model that adheres to your assumptions. And so for that, I don't immediately see the value of going like a more complicated virtual brain if you just want to test your analysis. So I see a use for this when you want to do something more complicated. So maybe do a more like a feasibility study. So in like an ideal case, can my method be used to extract information in this and this scenario? So maybe to explore the boundaries of where your method works and where your method stops working. So there it may be valuable. But
okay, generally
I would love to hear about this. But
so you're generally kind of happy with the simulation models that you have now. Lawrence and Etienne, do you want to say a word on this? So the use of more biological realistic models to test the tools?
So I agree with Marijn. It really depends on what your assumptions are, what you want to test. So if you want to just do an ERP study and you want to uncover with your pipeline what the true ERP is, then that is all you need to simulate, a true ERP. So Siriga, for example, does not simulate, like spontaneous synchronization of single neurons firing that then combine to get an ERP. That's too much detail for that particular question. You can just say, okay, my ERP has this latency, and now my method should uncover this latency. So for many of the EEG methods, which also go back to this linear assumption, if you want to test these, then a simulation framework that has this same linear assumption, I think, is appropriate. But more realistic models can be definitely useful for other types of questions. So if you want to simulate realistic resting state EEG with network connection and all these lower level field potentials, then you need a different model, and then the virtual brain, of course, would be a good choice. Yes.
Okay, thank you. Etienne, or you can say yes or no. You can skip the question if you wish.
Maybe.
Virtual brain, yeah or nay?
It's a discussion.
Yeah, okay. So maybe the other question, the other topic that I had in mind, going back also to the topic of this session, which is coded tools. So one of the key issues now, I think, for the next years, will be trying to develop pipelines that integrate multiple softwares. For example, that integrate software that come either for different modalities, such as MRI, EEG, functional connectivity, and so on. And then maybe even different coding languages, MATLAB and Python. So Marian mentioned this in his presentation. So you briefly mentioned NiPipe. Can you give a word on this, and then we'll have the word from the other two speakers?
And Jupyter also.
And Jupyter, yeah, that's true. So you can tell a bit, Etienne, about Jupyter. I will talk a bit about NiPipe. So what NiPipe does it creates Python wrappers around all these different tools. So so for example, if you have for MRI, if you have the FSL program, or you have the SPM MATLAB toolbox, then NiPipe will provide sort of a Python wrapper around this. So you can have a single Python script where you can call like the SPM and the FSL functions, what are written in. And the downside of this is that it adds a new layer of complexity to your data analysis pipeline. You have this layer in between. But when you set it up right, it's actually really, really nice, because it will also do sort of build tools, so things that will keep track of your intermediate results, and it will not recompute something you already computed. And so it's actually a really powerful tool to construct your entire data analysis pipeline. And then the different steps don't have to be just Python scripts. They can be almost anything that you want.
Etienne, can you add something? Do you want to add something?
Well, I didn't really mix the languages. Usually, I really try to find pure Python software and to stay within Python, and usually it works. I usually don't really need to switch frequently to MATLAB. That say, I know that with Jupyter you can define some cells, for example, that are going to execute R code, for example. I have seen once a notebook that was also compatible with MATLAB. I remember that it was terrible, but it was something like 5 or 6 years ago. But I know it's possible. And yeah, this is nice, but I think it's even better if we could have all the tools we need in the same language and spend more time writing code and spend less time writing code and more time analyzing your data.
Yeah, I agree with you there, Etienne. The best way is to just stay within a single language, and hopping between these languages, it adds complexity, and complexity is our arch nemesis in data analysis. The more complex our analysis becomes, the more likely we're going to make mistakes. And if you're going to integrate to this, is like a nice source of creating all kinds of weird stuff. And yeah, this is weird. It's a bit scary to do this.
And there is so much Python package you can find on GitHub that I think it's almost impossible to not find what you really want, in fact. So
there is a comment from Karim Gerbi talking about NeuroPython, and there is a link that you can have a look, which is based on NiPy. Lawrence, do you have a comment and a suggestion about this topic as well?
No suggestion, no. I would be asking for suggestions myself. I mean, I am mostly sticking to MATLAB at the moment, primarily because of EEG Lab. I think back in the days when I started in this field, M&E did not exist yet. But of course, I realized the downsides of using proprietary software, so I would definitely be interested in any good way that you combine different packages. I'm moving to a new lab now, so I have some time to reconfigure my workflow also, but I don't know of any way to easily combine different languages at this time, rather than just saving your intermediate results and then switching. But this is, as Marijn said, is not nice. It's not it adds complexity.
That's true.
Okay, so I think we are done. Now, so I thank you very much for all the speakers. And I wish I had all these tools 20 years ago when I was doing my PhD, but unfortunately it didn't happen. And so now it's time for there is the poster session, and there is virtual, so real beer or virtual and real discussion. So you can relax a bit. And congratulations and thanks again for the presentation. And I give the word to Clément, who is doing.
Thanks to you guys.
Thank you.
Thanks to you. Thanks to all of you, actually. I don't know if you hear me. Is that right?
Yeah.
Okay, great. So please don't leave your screen, actually. So as Andrea was mentioning, we have this very nice virtual world to explore once again, where you will be able to find our 57 or 58 posters in the poster room. Sorry. And we have also the pleasure to announce you the presence of a very specific DJ, the musician of mine, and he accepted to be the DJ for our social event tonight. So feel free to go through feel free to go there and to enjoy the music. You're gonna be able to find dance floor where you can press the Z button and show your dancing abilities. Anyway, I shut my mouth and I leave you in Crowdcast so that you can reach Gather Town from the link just below my face. We hope we're gonna see you all tomorrow for a very nice third day of conference. See you guys.